Welcome back to Dr. Liu's VCE Biology. Today's topic is disease challenges and strategies, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. MEIS is a viral disease likely originating in bats before jumping to camels via saliva. Humans typically contract the virus by consuming camel meat or milk. Once in the human population, the disease spreads through close contact and respiratory secretions, including coughs and sneezes. Currently, no specific antiviral treatments or vaccines exist for the MEIS pathogen. To break the chain of transmission from animal to human populations, we must rely on preventative measures. Heat acts as a molecular disruptor, altering the shape of the virus's proteins and lipids to transform a dangerous pathogen into harmless biological debris. Consequently, camel milk must be pasteurized, and meat thoroughly cooked before consumption. Europe has seen a recent resurgence of measles, a highly infectious virus transmitted through respiratory droplets and close contact. Although a vaccine has been available since 1963, leading scientists in the 1990s to believe elimination was possible, outbreaks have returned. This goal was based on the fact that the virus has no animal reservoir and that vaccination provides lifelong immunity. However, maintaining herd immunity requires a very high vaccination threshold, approximately 95%, to prevent the virus from spreading. Therefore, option A is the most likely cause of the resurgence. Reduced vaccine uptake during and after the COVID-19 pandemic has allowed herd immunity to erode, triggering new outbreaks. While option B, masking, and option C, improved programs, are helpful interventions, they do not explain the current rise in cases. Finally, option D is incorrect because contact with animals does not induce measles outbreaks, as the virus is exclusive to humans. A is incorrect, because an antibody is composed of 4 polypeptide chains working together. Its highest hierarchical level is quaternary structure, not tertiary. B is incorrect. Codons and nucleotides are components of DNA and RNA. As a protein, an antibody is composed of amino acids. C is correct. Monoclonal antibodies are proteins. All proteins are polymers built from amino acid monomers linked by peptide bonds. D is incorrect. A standard antibody consists of 4 polypeptide chains, 2 heavy chains, and 2 light chains, connected by disulfide bridges. YB is correct. In an autoimmune disease, the immune system mistakenly attacks the body's own cells. Monoclonal antibodies used in this context are designed to suppress or block the specific parts of the immune system, like inflammatory cytokines, that are causing the damage. Why the others are wrong. A. Attaching to viral infected cells describes an antiviral response, not an autoimmune treatment. C. Signaling T cells to attack self cells would make the autoimmune disease worse, not treat it. D. Increasing the activity of B cells would likely increase the production of harmful autoantibodies, which is the opposite of the treatment goal. The correct answer is C. Receptors on the CAR T cells must bind to a specific antigen on the cancer cells. Why C is correct. The diagram shows that CAR T cells are engineered with a chimeric antigen receptor CAR. For the treatment to work, these receptors must specifically recognize and bind to antigens on the surface of the cancer cells to initiate the killing process. Why others are wrong. A. The inserted gene codes for the receptor CAR that stays on the T cell surface, not for the production of free-floating antibodies. B. CAR T cells are added to the patient's system to target cancer. They do not replace all other T cells in the body. D. T cells kill target cells by releasing chemicals to induce apoptosis. They do not act as phagocytes to engulf cells. Helicobacter pylori are bacteria that infect the stomach lining. H. pylori are commonly treated with antibiotics. Some H. pylori are resistant to these antibodies. A combination of antibiotics is often recommended. Without proper treatment, H. pylori infections can continue and lead to stomach ulcers and stomach cancer. The graph shows how H. pylori responded to 5 different antibiotics over 2 time periods in patients who were infected. The correct answer is A. Amoxicillin and tetracycline. Why A is correct? Looking at the bar graph, amoxicillin shows 2 very short bars of equal height, approximately 2%, for both time periods. Tetracycline shows no bars at all, indicating 0% resistance in both time periods. Therefore, these two show no change in the percentage of resistant bacteria. Why others are wrong, B, C, and D. These options include antibiotics like clarithromycin, metronidazole, or levofloxacin, all of which show visible changes, increases, or decreases, in resistance between the 2009 to 2010 and 2011 to 2012 periods. The correct answer is C. Use of only metronidazole and clarithromycin may increase the occurrence of ulcers and stomach cancers in the human population. Rationale: the text states that without proper treatment, which becomes difficult if bacteria are resistant, H. pylori infections lead to ulcers and stomach cancer. The graph shows that resistance to metronidazole and clarithromycin is high and increasing. If these drugs fail due to resistance, more patients will remain infected, increasing the risk of those complications. Coxiella burnetii, a species of bacteria, can produce a disease called Q fever in humans. Humans can be infected when coming in contact with animals carrying C. burnetii. Animals that carry the bacteria include cattle, sheep, and goats. Person-to-person spread of the pathogen is unlikely. Symptoms of the disease develop 2 to 3 weeks after exposure to the bacteria. To determine if a person has the disease, a sample of blood is taken and tested for the presence of either antibodies against Q fever or the presence of C. burnetii DNA. Helper T cells act as the coordinators of the adaptive immune system. Their involvement follows a specific sequence of events. Any 3 of following items: initial activation. Helper T cells are first activated when they recognize and bind to specific antigens, such as those from C. burnetii, that are displayed on the surface of an antigen-presenting cell, APC, such as a dendritic cell or macrophage. Chemical signaling. Once activated, these cells secrete cytokines, which are chemical messengers that signal and recruit other immune cells to the site of infection. Expansion of the army. They facilitate the activation of cytotoxic T cells, which seek out and destroy infected host cells, and the activation of B cells, which differentiate into plasma cells to produce specific antibodies. Long-term protection. A portion of the activated helper T cells will differentiate into memory T helper cells, which remain in the body to provide a faster and stronger response if the same pathogen is encountered again in the future. Crucial distinction for students. Note: it is important to remember that helper T cells are not phagocytes. They do not hunt, collect, or present antigen fragments themselves. Instead, they rely entirely on APCs to show them the processed antigens before they can begin their work. To earn the full 2 marks for this question, an answer should connect the process of PCR to the purpose of identification. Mark 1. the process PCR, is used to exponentially amplify a specific segment of DNA from the burnetii pathogen found in the patient's blood sample. Mark 2. The identification. This creates enough DNA material to be visualized or analyzed, for example, through gel electrophoresis, allowing scientists to confirm the presence of the pathogen's unique genetic signature. You can choose any one of the following points to secure the mark: subclinical or mild symptoms. Many people may not feel sick enough to see a doctor, resulting in people not being tested for the pathogen. Poor timing of diagnostic tests. Testing may occur early in the onset of the disease, before antibodies are produced, or months after the onset when antibody levels have dropped too low to be detected. Asymptomatic nature of incubation. The disease can have a long incubation period, which means individuals are simply unaware they are infected and do not seek medical care. Economic barriers. There is a significant expense associated with collecting and analyzing blood samples from individuals, which can limit widespread screening and identification. To help you secure that one mark, you just need to provide one clear, non-vaccine-related strategy. You can use any one of these specific examples for your response. Public Health Administration, implement mandatory reporting of cases to health authorities, or enforce the isolation or quarantine of infected individuals to prevent further human-to-human transmission. Workplace safety and education, introduce education programs or disinfection protocols specifically for individuals working in high-risk environments, such as those working with livestock or in abattoirs. Animal management, manage the source of the pathogen by screening animals for burnetii, culling infected animals, or restricting the movement of animals from areas where the bacteria are prevalent. Low community risk. Unlike diseases in the NIP, Q fever has minimal person-to-person spread, meaning it poses a low risk to the general public. Targeted population. The risk is concentrated in specific occupations, like animal handlers, so community-wide herd immunity is not required. Economic cost. The high cost of vaccinating the entire population is not justified by the low number of infections outside of high-risk groups. Adaptive immunity and helper T cells coordination. Helper T cells are activated by antigen-presenting cells to release cytokines and coordinate the immune response. Effectors. They activate B cells, antibody production, and cytotoxic T cells cell killing, while forming memory cells for future protection. Q fever. Diagnostics and control detection, PCR, is used to amplify burnetii DNA for identification via analysis like gel electrophoresis. Barriers. 90% of cases are missed due to poor test timing, low awareness, or high costs. Government strategy. Spread is controlled via mandatory reporting, animal screening, and worker disinfection protocols rather than mass vaccination. Public health policy. The NIP Inference. Q fever is excluded from the National Immunisation Program because it lacks person-to-person spread and affects only specific high-risk groups. Logic. The government must balance the high cost of universal vaccination against the low community risk. Advanced immunotherapies, monoclonal antibodies, targeted proteins used to treat cancer or suppress autoimmune responses. CAR T-cell therapy, engineering T cells with specific receptors, CAR, to recognize and bind to antigens on cancer cells to destroy them. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.